So with the permission of the committee, I'm going to present the results of my doctoral thesis, which title is How to Understand Sensory Neurons from Structure Function Studies to Nanotechnology Development. Why do we want to understand sensory neurons? Why these sensory neurons are important for us? Well, we human beings, as any other living being, need to interact with our environment in order to survive. So we need to be able to collect all the information that we're constantly receiving if we want to survive. And to do so, we have these specialized neurons called sensory neurons that form our peripheral nerve system. And this peripheral nerve system is something like a Batman suit that is constantly receiving information about what is happening outside in the outside world and what is happening inside you. But it's not actually, it doesn't look like this, it's something more like this. So it's, you can see here all these sensory neurons innervate your whole body, creating this net of nerves. But what kind of information can we collect, or can these sensory neurons collect? And what I want you to do now is a little experiment. So I want you to think just right now, what kind of stimuli are you sensing now? So I will give you like a few seconds to think about that. Okay. So yeah, I'm quite sure that you've guessed these 5 different sensory modalities. There are the well-known 5 senses. So vision, hearing, smelling, taste, and touch. But there are many other things that we can sense. We can, for instance, sense the temperature. So we know if it's warm here, if it's cold. And we can also feel pain. And in this doctoral thesis, we were mainly interested in understanding the process of pain sensation or nociception. And the sensory neurons specialized in this sensory modality are the 3 nerve endings, and they're responsible for sensing the noxious stimuli, and they make us feel pain. But why do we want to study pain if we don't really like pain? Even if we don't like it, pain is very, very important for us. It serves a very important physiological role that is serving as a warning signal. So pain is teaching us to avoid dangerous situations. So for instance, we learn not to touch something that is very hot because we will get burned, and we feel pain because getting burned is dangerous. But also, when pain doesn't serve this warning signal meaning, it can become a problem. And in fact, chronic pain is a pathological problem that is a worldwide health problem. So if we want to be able to treat pain, or this kind of pathological pain, we first need to understand the whole process of pain transduction. What we know now about pain transduction is that everything starts with an noxious stimuli that is sensed by our peripheral nerve system in the endings of these sensory neurons. So these sensory neurons express these specific receptors called nociceptors that are responsible for sensing these noxious stimuli. When they are activated, they generate an electrical signal or an action potential that will travel along the whole sensory neuron to the spinal cord, where the cell body of these neurons lies, and then from there up to the brain. And then the brain will make the adequate decisions to avoid the noxious stimuli. But if we want to understand this whole process, that is a very, very complicated process, it's a good idea to start studying these nociceptors. So what we wanted to do is to know more a little bit, to know more about how these little machines work. So this is like understanding how an engine works. So we need to know how these little pieces and parts make the whole engine to do its function. But first, we need to know which are these molecules. And they are usually ion channels or proteins coupled to ion channels. And two of the more important ones are these two, that belong to the family of the transient receptor potential channels. And they are called transient receptor potential binary 1 channel, or also is known as the capsaicin receptor, which is the spice ingredient of chili peppers. And also the transient receptor potential melastatin 8 channel, that is also known as the menthol receptor, which is the refreshing agent of mint. But these sensors are also activated by different stimuli, like for instance, noxious temperatures in the case of TRPV1, so temperatures above 42 degrees Celsius. And in the case of TRPV8, it's also activated by temperatures below 28 Celsius degrees. From an electrophysiological point of view, these channels are inward rectifying channels, and they are non-selective cation channels that causes the depolarization of the membrane upon activation. From a more structural point of view, these channels form tetramers, and each of the 4 subunits have a predicted structure of 6 transmembrane segments, with the pore region formed between by the loop between the S5 and S6 transmembrane segments, and with intracellular N and C terminus. And the difference between the members of the family lie in the sequence of these intracellular domains. But there is an important region called the trip domain that is shared among different members of the family, in particular TRPV1 and TRPV8 channels. They have this characteristic domain that is being involved in the tetramerization of the channel, but is also being defined as a very important region of the protein for the regulation of the channel opening by different stimuli. So we define the first objective of this doctoral thesis as the study of the function of this trip domain in trip hypnosis sensors. To do so, we follow a direct mutagenesis strategy. So this is the TRPV8 channel. You can see here this is the X6 transmembrane segment, and this is the trip domain. And there is a region here that is called the trip box that is even more conserved than the trip domain. And we were interested in studying this linkage region between the X6 transmembrane and the trip box. So what we did was to obtain different chimeric channels between the TRPV8 channel and the TRPV1 channel, so menthol and capsaicin. As you can see here, we were sequentially and cumulatively adding residues to obtain these 3 different chimeras from chimera A to chimera C. After that, we were analyzing the function of these chimeric channels by transfecting cells with the cDNA, and then using particle technique in whole cell configuration. So regarding the first of the chimeric channels, when we transfected the cells with chimera A channels, we found that cells were dying more than in the control situation. So we studied the hypothesis of these channels being toxic to the cell because they were constantly open, so calcium was entering the cell constantly, and that was the cause of the cell death. So to test this hypothesis, we cultured the cells in the presence of a channel blocker, and then we applied this protocol, so we were washing the blocker, then we were adding it again. And we were measuring this in whole cell configuration at -60 millivolts, which is a voltage where the channels would be closed, at least in wild-type channels. And indeed is what we observed. So as you can see here, there was no change in the current, so there was no current in TRPV8 wild-type channels. But when we repeat that experiment in using chimera A channels, we found that upon removal of the blockage an inward current was observed. That was confirming our hypothesis of the chimera A channel being constitutively active. But afterwards we were wondering, because only 3 amino acids are different from the tryptamate wild-type channel and chimera A. So we were wondering which one of them was responsible, or how many of them were responsible for this effect. So what we did was to obtain the single point mutations and again repeat the same kind of protocol. And what we found was that only mutation in the position 991 was responsible for the observed phenotype. After that, we studied the function of the other 2 chimeric channels. So as you can see here, both of them, chimera B and chimera C, even in the absence and presence of 1 millimolar method, were showing voltage and method regulated activity, in contrast to chimera A. So these are the results, current results from the step protocol, voltage step protocol from -100 millivolts to +240 millivolts. As you can see here, both of them were responding to both stimuli. Even chimera C seemed to be less responsive. We analyzed all this data by obtaining the normalized conductance to voltage relationships, as you can see here. In order to be able to compare the effect of the different mutations, or the effect of the different mutations, we were utilizing a parameter called the voltage needed to reach half of the maximal activation. So voltage needed to reach 0.5 here. And these are the values that are shown here. And this value can be related to the energy that channel that needs to be applied to the channel to open it. So as you can see here, in the case of chimera C, as we were expecting, there was a right shift of the voltage to more depolarized voltages, which means that higher energy needs to be applied to the channel for it to be open. In the case of chimera B, in the presence of menthol, the response was very similar to tryptamate wild-type channels, but in the case of voltage, there was a favored opening of the channel in the case of chimera B. However, the most interesting result of all these experiments was to find that even chimera A was constitutively active. Chimera B showed a regulated behavior similar to tryptamate wild-type channel. So, because only 5 different amino acids were added to chimera C to obtain chimera B, we decided again to try to find which ones were the responsible amino acids for this recovery of the regulated activity. So we did again the single point mutations of these 5 different amino acids in a chimera A background, and we found that mutations in position 987 and 988 didn't have any effect, so they showed similar behavior to chimera A. But in the case of positions 986, 989, and 990, they showed a similar behavior to chimera A, with a slightly more favored opening for voltage and method application. So, in conclusion, we know that these 4 different amino acids are very important to define the energetics for channel opening. So what we did next was to try to locate these residues in a structural context. So we developed this structural homology model based on the model for T1 channel. And as you can see here, these are the 4 different subunits. And in red, there are shown the trip domains. And this is the X6 transmembrane segment. So this is the region that we were studying. In the case of the residue in position 981, this tyrosine, we found that it was lying in a hydrophobic pocket, as you can see here, formed by the trip domain of a neighbor subunit. And we found that in the open state of the channel, this interaction was lost. So adding aspartic acid like the mutation in chimera A here, could somehow mimic the open state of the channel. So that's why the chimera A was constantly open. In the case of the other three residues, they were found in this mess. So they were establishing different interactions between the S4-S5 loop of the same subunit and the trip domain. So again, all these interactions could be really important for establishing, for maintaining the channel in the closed state, and again, defining the channel energetics for opening. So in conclusion, we demonstrated the fine-tuning of this linked region between the X6 and the trip box in the trip domain was very important to maintain the characteristic properties of the wild-type channel. And also we found that interactions between this part of the trip domain and the S4-S5 loop and the core gate are very important for defining the behavior of the channels. And indeed, they could be potential targets for the design of new analgesics. So now we know why we want to understand sensory neurons, and now we know what structure function studies means in the title of my thesis. But what about nanotechnology development? So from now on, I will be talking about this second part of my thesis. But first of all, what is a nanotool? Well, a nanotool is, as its name indicates, a tool that has a nanometer size. I just wanted to know, or to give you an idea of what a nanometer is. So it's this 0.00001 millimeter. So it's something, it's just having a very tiny screwdriver. But why do we need nanotools? There is no doubt that studying the structure-function relationships of these nociceptors give valuable information about how we can sense noxious stimuli. But if we really want to understand the whole pain transduction mechanisms, we need to study these proteins in their native environment. And the problem is that when we try to study them in the whole cellular context, a lot of new players come to the game. So there is not only, we need to study not only this protein, but also the proteins that are interacting with it. It can be interacting with the cytoskeleton. Also the lipids in the membrane would affect the behavior of these channels. So we need to be able to study it in this environment. But it becomes even more complicated because all these hundreds of molecules are not randomly distributed in the cell. They are occupying specific positions at specific times. So if we really want to understand the function of these channels in the native cells, we need to have tools that will allow us to study this differential expression. So that's why we need nanotools. In particular, in the case of ion channels, the distribution in the membrane of cells in sensory neurons is being defined as a very important way of modulating cellular behavior. And in particular, in the case of sensory neurons, it seems very important to have a way of activating these channels only in the periphery of the sensory neurons, because that will give us the possibility of having a more physiological model to study pain. Because till now, the experiments that we used to do are with the whole neuron, so we are stimulating also the cell body, but the cell body of these neurons is in our spinal cord. So having the possibility of studying these or stimulating these channels in the periphery of the sensory neurons would be very important. So to do so, we decided to define our second objective of this doctoral thesis as the development of a nanodelivery tool to locally stimulate TRPV1 channels in the membrane of sensory neurons. So to do so, that was the main idea. So we decided to have a pipette that would be filled with capsaicin and would be delivered in different points of the sensory neuron while we were recording channel activation. But we want to make it a very local delivery, so we were using nano pipettes, and this is where the nano comes. And we were using these borosilicate pipettes with a radius around 15 nanometers. Then we wanted to make it quantitative. So we needed to take into account all the different parameters that will be affecting the profile distribution of the capsaicin outside the pipette. So we, of course, need to know the concentration that we are putting inside the pipette, but also we need to know the physical-chemical properties of the solutions. We also need to know the properties of our molecule. Of course, the delivery force that we are applying to push out the molecules. Also, the geometrical properties of the pipette will affect how this profile distribution will be. And a very important parameter that we need to take into account and we need to be able to control is the distance between the nanopipette and the cell. And this is not trivial. And to do so, we utilize the SICM technology, or scanning ion conductance microscopy technology. And this technology is based on this effect. That is the reduction of the current that happens when we approach pipette to an ion permeable surface. So this technology utilizes this reduction in the current as a feedback control of the position of the pipette. So considering all these different parameters and having the option of controlling the distance to the pipette, between the cell and the pipette, we obtain this analytical expression that will give us the concentration of our molecule as a function of the distance on the surface. And as you can see here, this is the initial concentration that we're putting inside the pipette. This is the distance between the pipette and the cell. This is again the distance on the surface. And this is the total flux of solution leaving the pipette. And it depends on the delivery force that we are applying, either pressure or voltage. So we confirm the utility of this analytical expression by computational simulations. This is the result of these simulations. As you can see here, this is the profile distribution of molecules outside the nanopipette. And this is the color code of the concentration. So as you can observe here, this is a very local concentration, and this is a very local delivery tool, because the concentration is dropping very, very fast at the tip of the pipette. And you can see here that the simulations, even when the delivery was made by pressure or by voltage, they coincide with the theory. So we could use this analytical expression to calculate the profile distribution of our capsaicin. Then, also using these analytical expressions, we can define how the profile distribution can be controlled just by modifying different parameters. So, just by modifying, for instance, the distance between the pipette and the surface, you can see how we can modify the profile distribution of the molecule being delivered. So, as you can see here, the closer we are to the surface, the more narrow is the application, and the higher the concentration that we can reach at the very top of the surface. But because we wanted to be able to deliver our capsaicin in different parts of the sensory neuron, we designed this protocol. So we used confocal microscopy in combination with SICM. And what we were doing was to loading our cells with a calcium-sensitive dye. So we will see activation of TRPV1 channels by an increase in calcium concentration. That will be an increase in fluorescence. And we were aligning the pipette with the laser, so we will be sure that we record the fluorescence just under the point that we were that it was stimulating. And at the beginning, we were obtaining these raster scan images, so we were just scanning like this the field that we wanted to study. And in this way, we were obtaining the coordinates of all the points in this field. And then with the SICM system, just by clicking in the points that we wanted to apply the capsaicin, everything was done automatically. So the kind of protocols that we were applying was we were applying a step voltage protocol with the voltage that we wanted to deliver. The higher the voltage, the higher the capsaicin, the higher the concentration that will be achieved. And then we were recording the ion current through the pipette as a control of the delivery to see if, for instance, the pipette was blocked by something. We were also recording the position of the pipette at all times. So at the beginning, the pipette was hopping from the top to the cell, 5 microns, as you can see here. But as soon as the delivery started, it was stopped at a fixed position, usually defined by us, usually 300 nanometers in our case. And then, of course, we were recording the fluorescence. As you can see here, an increase in fluorescence was happening when TRPV1 channels were stimulated. And as you can see here, the higher the voltage that we were applying, the higher the response that we were getting. And because we were applying more capsaicin and we were also covering a wider area in the cell. And what we did was to measure the slope of this linear part of the fluorescence response as a measurement of different voltages. And we were able to obtain these dose response-like curves, as you can see here, at different points in our sensory neurons. So it's very important that we were able to independently stimulate these different points. Okay, let me see. Let me explain this. This is one neuron, this is the cell body, and these are the different dendrites. You cannot see it properly, but they are there. So this is a control point outside the neuron, and these are different points. And in this case, only these 1, 2, and 3 points were responding to capsaicin application. And this is because probably these other points belong to a different neuron that doesn't express TRPV1 channels. But as you can see here, we were able to independently stimulate these 3 points that are very close. So this is also confirming that our delivery is very local. And then we were even able to record different behaviors in different points, which can be related to a different channel distribution or even different channel properties. So these are the responses in these 3 different points and different voltages applied, which translate to different capsaicin concentrations. So in conclusion, we developed analytical expressions to calculate the concentration distribution profiles of delivered molecules through glass nanopipettes. And we also developed an automated system based in confocal microscopy and the SICM technology to deliver the TRPV1 agonist capsaicin to sensory neurons targeting different subcellular areas. Again, with the same idea of the development of new nanotubes to be able to study sensory neurons or ion channels or even processes happening in sensory neurons in the native environment. One field that could potentially benefit a lot from this approach is the measurement of oxidative stress. And oxidative stress is the overproduction of reactive oxygen species in the cells, and this oxidative stress is being related to different pathological pain conditions. And the challenge of measuring this oxidative stress in cells is that these reactive oxygen species have a very, very short lifetime. They are produced very locally in the cell and in very, very low concentrations. So potentially, nanotools are very handy to study these processes. So, last but not least objective of my doctoral thesis was to develop an intracellular ROS nanosensor. And to do so, again, we used nanopipettes, but in this case they were quartz nanopipettes, and we paralytically deposited carbon inside this nanopipette to generate these carbon nanoelectrodes. They look like this. So this is, again, a nanopipette around 15 nanometers diameter. And the idea was to further functionalize these carbon nanoelectrodes with Prussian blue, that is an electrocatalyst for the reduction of hydrogen peroxide, which is one of the most dangerous ROS species. So the idea was that this Prussian blue will catalyze the reduction of hydrogen peroxide to water by the oxidation of the iron 2 in the complex to iron 3. And then this iron 3 will be recycled to iron 2 by applying a negative voltage through the nanoelectrode. And then this current that we will apply will be equivalent to the amount of hydrogen peroxide in our sample. So after functionalizing our nanoelectrodes, we studied the sensitivity to hydrogen peroxide of them. So this is the negative control, it's a bare carbon nanoelectrode. As you can see here, neither in the activation solution, that is the solution that we utilize to prepare to deposit the Prussian blue film in the nanoelectrodes, nor in the presence of 5 millimolar hydrogen peroxide, we were able to record any current, which is very good because this is our negative control. Then, when the nanoelectrodes were modified with Prussian blue, in the activation solution, you could see the appearance of two different peaks that are related to the oxidation and reduction of the Prussian blue. So they are indicating that we actually deposited a film of Prussian blue there. And then in the presence of hydrogen peroxide, there was an increase in the negative current that was related to the amount of hydrogen peroxide in our sample. So by recording this value of the current in different hydrogen peroxide concentrations, we calibrated our different sensors we prepared, and we calculated an average sensitivity of around 250 amperes per mole per square centimeter, which is a very good sensitivity for this kind of sensors. And afterwards, because we aim to have an intracellular ROS nanosensor, we were using these electrodes inside cells. So these are the kind of experiments that we were doing. So this is our sensory neuron before penetration. This is after penetration. This is after 20 minutes of PMA application. And PMA is an inductor of ROS production because we wanted to measure ROS. And this is the poor cell after the retraction, which is kind of okay. And this is after the addition of 1 millimolar hydrogen peroxide that we were adding to test the sensitivity of our electrodes. So, regarding the measurements that we did in these kind of experiments, this is again the negative control, so we didn't see any difference upon penetration or retraction of our nanoelectrode if it was a bare carbon electrode. But then when we were using our Prussian blue modified electrodes, we were observing these current spikes after penetration and also sometimes after retraction of the electrode. But unfortunately, we weren't able to see any differences in the current after PMA application. And this is probably due to the very low detection limit of our electrodes. because the concentrations that we are expecting inside cells are very, very low. Because indeed our electrodes were sensitive to hydrogen peroxide, but not enough to detect the concentrations inside the cell. So in conclusion, we successfully fabricated plasma blue modified carbon nanoelectrodes with hydrogen peroxide sensitivity, and we utilized them to perform intracellular recordings in sensory neurons, showing that cellular viability was maintained, but we need to further improve the detection limit of these nanotubes if we really want to be able to detect ROS inside cells. So this is all. I would like to thank supervisors Antonio and Yuri, and also Gregorio, Peter and Francisco for their scientific contribution to this thesis, and also to the Ministry of Education for funding my PhD studies, and also a todos vosotros por haber venido a verme.
Thank you very much.